So as August Weizmann suggested, the germ cell theory of heredity in 19th century, germ cells are very unique cells that can go over the generations by maintaining the ability to generate whole organisms, unlike somatic cells that can finish their role at the end of the generation. Germline is a line of heredity. However, they also reset each generation and undergo very complex development. My question here is, how is germline maintained over the generations? What is the role of this complex development for heredity, particularly in us human? The functional form of germ cells in human are egg and sperm. However, they come from the common precursors called primordial germ cells. We often call them PGCs. Human germ cells become functional and mature at puberty around 10 to 15 years old. But the development initiates way before in the uterus as PGC specification only week 2 to 3 after the fertilization. In the tiny embryo, less than 1 millimeter, called epiblast. So here is the human epiblast looks like. You can see epiblast lying as a flat layer, and you can see a gastrulating cells that comes from the epiblast that will form the body later. And the BMP signaling from the surrounding tissue induce primordial germ cells, PGCs, that would express specific markers such as nano3 or TNAP. The investigation of human PGC specification is extremely challenging since this particular stage of the human embryo is highly inaccessible. And the primary model for mammalian development mouse shows a very different morphology of the epiblast. You can see an elongated epiblast here. And importantly, an extra embryonic ectoderm, that is a critical source of BMP4 signaling to specify PGCs, is not existent in human epiblast. On top of that, we didn't have a robust experimental model to investigate the question. So here we took these experimental strategies. So when PGCs are specified, they migrate through the body and reach the gonadal tissue, the future ovary or testis, where PGCs become gonadal PGCs and undergo dynamic epigenetic programming, such as global DNA demethylation. So we can access to these gonadal PGCs from the aborted fetuses, such as week 7. So we investigated and characterized these cells as a reference for the research on PGC specification. And importantly, we can make use of human embryonic stem cell culture that is derived from the earlier stage of the embryo called blastocyst, and addressing how human PGCs are specified. So first, we made a nano3 reporter and established a detection system for TNAP to be able to detect PGC specification from human embryonic stem cells. The first attempt has been done by adding the critical factor BMP on the conventional ES cells and form 3D aggregations. However, also previous reports suggested that this was really efficient. The PGC population that was expected to appear here by flow cytometry analysis, expressing both markers, will not appear efficiently. And the resulted cells showed aberrant gene expression profiling. So if we looked at the in vivo PGC specification from blastocysts carefully, there's an important intermediate step is there, we can call it germline competent state. So now the idea is to induce this germline competent state from conventional ES cells, then induce PGC specification. By searching a number of conditions, so we found that the ES cells cultured with these 4 inhibitors shown in here had an excellent competency to induce PGC fate up to 60% over 4 days. So this experiment suggests that in vitro PGC specification is such a sensitive method that requires a step-by-step protocol induction of appropriate germ cell precursors. So this robust specification system had a powerful platform to identify a number of interesting and important factors that are related to PGC specification. So I will introduce a little bit about the factor called SOC17, which we found that the factor expresses very early time point of the PGC induction from the germline competence state. To address the function of SOC17, we deleted the SOC17 locus in the genome and the human ES cells and induced PGC specification. So that was striking to see that the PGC specification was completely abolished in the absence of SOC17. And on the other hand, we tried, instead of adding BMP to induce PGC specification for 4 days, we introduced the expression of SOC17 and BLIMP1. This is other important factor for PGC specification, was robustly induced human PGC specification only in 2 days, independent of BMP signaling. So this suggests that SOC17 is extremely important for human PGC specification. So this is interesting in other aspect as well, since we know that mouse PGCs do not express SOC17, and they express SOX2 instead. And the following research identified that the monkey and pig, which also show a flat disc-like epiblast, express SOC17 in their nascent PGCs. And on the other hand, rat also shows an X-cylinder epiblast, shows SOX2 expression in PGCs instead of SOC17. It seems that the rodent and non-rodent in mammalian animals show or developed a different system to induce germ cell fate. So this is evolutionary point of view, very interesting. It also teaches us that we need to carefully take the animal model to study early human germline development. So this PGC specification in vitro system shows the peak around day 4, day 5. The longer culture didn't induce further development. Rather, PGC population started to decline after 6 days. So we characterize further this day 4 PGCs induced in vitro by having a global transcriptome data and plotted in the 3-dimensional graph. So here is the precursor germline competent cells, and this is plotted far away from somatic cells. And here's the in vitro induced PGCs. And this is in vivo gonadal PGCs from week 7. So we can see a very nice developmental trajectory from the precursor towards gonadal PGCs through in vitro induced PGCs. On top of the characterization of the global DNA demethylation profiling, we can speculate that the in vitro induced PGCs are equivalent to week 3 PGCs, which is before migrating towards gonad. So how far these week 3 early PGCs are from functional gametes that establishes in around puberty? So let's say puberty is 13 years old, and which is 678 weeks, adding up 38 weeks of development. The week 3 PGCs are 0.4% of the whole gametogenesis in human. So it seems to be a small progress, but it is important first step of the germline development. Considering particularly for the PGC specification, it was essential to have a stepwise developmental protocol. So the key question here is, do we need to culture germ cells for 13 years to induce mature gametes? We still don't know the answer. We might get the answer in 13 years. However, so there are some attempts to induce the further development using in vitro induced nascent PGCs by using by mimicking the in vivo situation developing further in the gonads, instead using a mouse embryonic gonadal somatic cells done by a Japanese group, Michinori Saito lab. And this is to mix the two cell types, human in vitro induced PGCs and mouse embryonic gonadal cells, to create a xenogenic ring-constituted gonads. So this system successfully induced oogonia using female mouse gonadal somatic cells, and by culturing for 11 to 17 weeks, which is 2 to 3 months. However, in vivo female human germline development, oogonia is differentiated around week 5, so which is 2 weeks after PGC specification. So it seems that this xenogenic system takes much longer to reach oogonia stage. On top of that, the meiotic entry of folliculogenesis that is happening followed by oogonia differentiation is not happening in this system. So similar attempts has been done using the male mouse gonadal somatic cells. So this case also takes 11 to 17 weeks to reach pro-spermatogonia cells. However, it reached a slightly later stage around week 9, showing mitotic arrest characteristics of prospermatogonia. So this is an exciting achievement, but also there's a limitation, as I said, like developmental timing, stage, or efficiency. And this PGC development is totally dependent on the unknown factor that is coming from mouse gonadal somatic cells. So here is a lot of technical challenges remain to reach true human in vitro gametogenesis. However, once we reach and having gametes-like cells in a dish, we can use it for in vitro fertilization to let them develop to the blastocyst, which can be used to derive ES cells as the next generation cells. So that will be ideal tool to incorporate the technique like genetic or epigenetic engineering, or using a patient-derived iPS cells. So this is really exciting in vitro model to understand molecular cellular mechanisms of human germline biology or reproduction and dysfunction. And this system will be extremely powerful for robust screening methods such as gene engineering or drugs to find a therapeutic target for germ cell related disorders. And also exciting to use this system to investigate the establishment or inheritance of genetic and epigenetic or cellular components such as mitochondria to see how this can impact on human health, diversity, or evolution. With that, I would like to show my acknowledgement, especially Azim Srani, who's been a great mentor, supporting this challenging research, and former and past lab members and the great collaborators. It's exciting scientific discussions, and the research has been not possible without the funding body. And thank you very much.